All right, I think we're going to get started. Thank you so much for coming. We really had no idea if anybody would show up, and we had no idea if we should actually go ahead, but appreciate you being here. And definitely appreciate that Rob was able to make it in today as well. We're lucky that you weren't flying in from somewhere. Dr. Beiko is an associate professor and Canada Research Chair in Bioinformatics. And I think that is worth noting given the constant message that we hear that there are not enough bioinformaticians anywhere, particularly in Canada. So it's great to know that we have this resource here in Halifax. We're very lucky. He's been with the Dalhousie Faculty of Computer Science since 2006. He spends most of his time trying to make sense out of large biological data sets, trying to uncover the ecological and evolutionary stories that have given rise to modern biodiversity. That's my phone, I'm sorry. His main focus is on microbiomes, groups of microbes that are found in a particular setting that interact with each other, consume resources, and generate products that can be beneficial or harmful to us. I'm sure you all know that. Before coming to Dalhousie, he developed algorithms and machine learning approaches to understand microbial function and evolution as a postdoc at the University of Queensland and a PhD student at the University of Ottawa. I should also mention, because a lot of you are actually new to our series, the way this works is that Rob will speak for about 40 minutes or so, and then we'll open it up for questions. Because we like to videotape these sessions so that people can see them after, when you do ask a question, I'm going to ask for you to wait for me to get over there with the microphone so that we can hear the question. It makes it a lot more valuable for those people who aren't attending in person today. And I think that's it. Please enjoy your time.
Thank you very much. So first of all, I would like to thank very much Sue and Terry for their efforts in organizing this. When they first asked me to do this back in September, I'm like, you're probably going to get nobody coming to this talk. You know, and here we are, right? So, but no, seriously, thank you all very much for making the effort in spite of the blizzard of 2014 to come in, and I do appreciate it. Okay? So just as a bit more introduction, it was mentioned that I am in the computer science faculty, and so you might have this question in your mind, what on earth is a computer scientist up here doing, telling me about bacteria and humans and so on? And so part of the reason is that I actually do have a biological background, and the other part of it is that the data sets that we get from these types of assemblages or whatever, you know, they're very large, right? They're huge, and so we need to think very carefully about what sorts of techniques, including computational techniques, we're going to analyze them, right? But in spite of that, I did want to start with the general overview of the microbiome and how things work and what we know and what we think we know, what we don't know that we know, in order to set the stage and for some people who may not be as familiar with the workings and the importance of the microbiome. So let me start here. No view is ever universally held in science. At the same time, I think it's fair to say that for a very long time, the general thinking around microbes, bacteria, was sort of had a focus on infectious disease, right? And so, you know, you think about bacteria as being harmful, we fight them with antibiotics or other means, and they really are something that needs to be fought off, right? But what's happened in the last, particularly in the last 10 or 15 years or so, is that this view has somewhat changed, and people have gained an appreciation not only of bacteria as pathogens, but also as important players in the general human ecosystem. So it's not just about them attacking us and making us sick. Really what we have are a diversified set of organisms that are involved in all sorts of interactions and do all sorts of processes that are beneficial to us. And some of these processes include interactions with the host, right? So this could be the intestinal mucosa, right? They interact with each other through various means, such as signals, right? They all have their roles to play. And the role of many of these tends to be more positive, right? And so we need to think about this and really appreciate how these systems work. This is not easy, as you will see. And so let's start with the definition. Okay, so the term microbiome was coined by Joshua Lederberg back in 2000 or 2001. And one thing that's nice about this, as with so many other things, is that there's an ambiguity about what the word means. So the first definition is the microbiome. And what this is the set of genomes that are associated with our microbiota. So you might want to look at all the genes that come from the bacteria and other organisms that live inside us. Okay? So this is really focused on the omics, the genetic information. Now, another way to look at it is the microbiome. Right? And so a biome is just the set of organisms that live in a particular setting. Right? And so the difference here is that we're not only thinking about the genomics, we're thinking about how that maps into diversity and function and ecology. So it's a more inclusive view. And this is the view we tend to take because it encompasses DNA sequencing and all of the other things that you need to do in order to understand what's going on in your gut or anywhere else for that matter. And so a few useful things to start with about the microbiome. Okay? The first thing to know about the microbiome is that there is no the microbiome. At any level of resolution you look, you'll find that there are differences, right? There's differentiations here, such that if you look, for example, at samples from the gut, you immediately see that the distribution, this is bacterial phylum classes, by the way, there's dominance by certain groups such as Bacteroidetes or Bacteroidea, whereas if you look at other body sites, you see very different patterns. So there is this differentiation, and this has a lot to do both with the ecology of the site where they live, as well as the interactions they have with other organisms in the mix. And so just to highlight this, excuse me, if you look at fecal samples, and I'm afraid the color's not showing up there, but fecal samples tend to have the highest incidence of members of the group firmicutes. Okay? And so one question to keep in the back of your mind are, what are firmicutes? Not just in terms of what their diversity is, but what sort of role they play. So we're going to keep coming back to this. So another thing to know is that the human microbiome fluctuates through Ting, okay? Or anyone else, for that matter, right? So what I'm showing you here is a study that was done a couple of years ago called Moving Pictures of the Human Microbiome. And what they did was quite impressive. For about, there are 440 days, every day, 2 individuals took a fecal sample, extracted the DNA from that fecal sample, and did sequencing in order to very minutely track the progression of different bacterial groups. And so what they found was, not only is it dominated by several groups in particular, they also found quite a bit of variation. You can see this no matter where in the room you're sitting, right? So let's take something really common like the Bacteroideaceae, right? This is a very common constituent of the gut microbiome. And so you can watch it fluctuate over time. Sometimes it's higher, sometimes it's lower. And so when it's lower, other bacteria are taking its place. So what does this mean, right? Another thing to know, which is central to a lot of the work that's going on in microbiomics right now, is that if you look at the microbiome and you know what to look for, you may be able to distinguish between healthy and disease states. And so here's an example, looking at, in particular, type 2 diabetes. There are many examples of this, inflammatory bowel disease, for instance, but in this case, it's looking at diabetes. And if you look closely, what you see here is for different groups of microorganisms, so Bacteroidetes, firmicutes, proteobacteria, so these important constituents of the microbiome. On the top, excuse me, the blue circles are observed frequencies from each of the number of individuals. They're sort of percent composition of Bacteroidetes. Okay? Those are healthy individuals. People with type 2 diabetes are shown with the green triangles on the bottom. The pluses show the incidence, right? And so even with this comparison, you can pretty quickly see that in diabetics, the frequency of beta proteobacteria is quite higher, Bacteroidetes is higher, and then some of these Clostridia are considerably lower. And so this is kind of a first suggestion of what's called dysbiosis, right? So the normal role played by our gut bacteria might be disrupted, and part of that disruption might be reflected in the different things that you find there. The other thing to know, and this is a frequently cited factoid about the microbiome, is that the human microbiome is very complex. Okay? And so, there have been many arguments about how many genes humans have, right? And prior to the sequencing of the human genome, there was a betting pool, and I believe everyone who placed a bet was too high. Right? Humans are complex, so they must have 50,000 genes. Well, the current estimate falls, I believe, somewhere between 20 and 25,000 genes. Okay, so that's for us Complex organism, complex development phase, lots of differentiated organisms, 25,000 genes. We compare this against the human gut microbiome. Now we're talking for an individual about 2 to 3 million genes from all the bacteria. And for most of the samples that have been collected from healthy individuals, typically you have 160 greater species for any given sampling time. Now species is in quotation marks because typically when you talk about these things, you're not exactly looking at species, you're looking at clusters based on some criterion of similarity, but we're just going to treat them as species, okay? Because they are, we're kind of assuming these are ecological units. And so just to kind of drill down here, because I told you things in broad scale, you can say, all right, here's the distribution, like the moving pictures story I showed you, right? And so there's all these different constituents, and you can say, well, what are each of these different constituents doing in the microbiome? And so the answer in a lot of cases is, well, we don't really know beyond maybe one function. And so let me tell you about the genus Rosaburia. And the basic thing that's going on here is that Rosaburia is a commensal bacterium that lives in our guts. So it's actually quite useful to us. What it does is it takes these various carbohydrates, such as starch and inulin and various other types of oligosaccharides, okay? Rosaburia says, yes, more please, right? It goes to the buffet of carbohydrates. And what comes out of that is a number of things, but of particular importance is this molecule butyrate. Okay? So Rosaburia is a butyrogenic organism. Why is butyrate important? Okay, well, there's many, many reasons why butyrate is important. For such a humble, simple little molecule, it's quite remarkable, actually. And so one important thing about butyrate is that it's a crucial energy source for colonic cells. Take this away, and the colonic cells get very depressed about things, and they tend to explode. Regulator of gene expression in our mucosal cells, right? So they induce changes, the butyrate does. Protects against some diseases, right? So these things are all tied up together. There's hypotheses about inhibiting cancer cell proliferation. And one very interesting thing is that it's been shown that the butyrate molecule can actually act to suppress growth of pathogens like Clostridium difficile. So this one simple molecule has a number of beneficial impacts. But here's the thing about it. So Rosaburia synthesizes butyrate and gives it to us in some sense. But it's not the only butyrogenic organism. There's actually quite a few others, such as Fecalibacterium, Ruminococcus, and Butyrivibrio. So there's all these different genera from different parts of bacterial diversity that all produce butyrate. Why? Why are there so many? What's the difference between them? Is there any difference? Right? Is there any difference we care about? Of course there's going to be variation in their genome, but does it make a difference to us? Is it okay to have Fecalibacterium or Rosaburia and you don't care? Or are there these differences beyond this sort of, this basic fundamentally important butyrate synthesis? Is there anything else that is of ecological importance to us? Right? And long story short, I don't think we really know yet. And so one of the things that's really interesting and important about the microbiome, why people are putting so much effort into it, is that unlike our genomes, right? Our genomes obviously influence our phenotype and can predispose us to certain types of conditions or diseases. Well, our genome is quite difficult to manipulate, right? People have tried and there's been some success, but it's really an intensive process. On the other hand, our microbiota, our other genome, right, is much easier to manipulate for better or for worse, because certain types of input can drastically change the composition of microbes that you have, whether it's in your mouth or your gut, and so on. And so just a couple of examples, which many of you will no doubt be familiar with. So here's a table from a nice review that was done a couple of years ago now. And basically what they're talking about is, if you start somebody on a particular diet, and then you move them to a different diet, okay, so you can see high fat, carbohydrate reduced, vegetarian diet, what is the impact on the microbiome? Is there any change at all? Yes, there is. And so you can look at all of these and you see these inscrutable genus names and class names and so on. And maybe you don't know what they do, but you can say, oh yes, they're going up, or they're going down, or they're unchanged. And so let me draw your attention to one in particular, which is this one, refined sugars, which we all know and love. I just had 2 refined sugar units before the seminar started. And so bacteria altered. Well, a couple of Clostridium, including C. diff, on average, increased in abundance. I think many of us, excuse me, have latent C. diff, but often held in check by some other organisms, such as the deuterogenic ones. But certain types of environmental influences can change that balance. A very, very widely publicized example of this is actually trying to take the microbiota from the gut of a healthy individual and then transferring that into an individual with dysbiosis, some sort of metabolic syndrome, or an infection such as C. diff, to try and remedy that infection. Okay? And so C. diff has kind of been the poster child of this because it really is a disruption in the normal microbiota. So this is a really interesting study, again published a couple of years ago, basically looking at mice. Okay, so healthy mice on the one hand, and then these what they call super shedder mice, which are infected with a particularly virulent strain of Clostridium difficile. So what you're looking at here is a principal components plot. Never mind that. Basically what it is a 2-dimensional summary of the microbiota in each of these organisms. Organisms that have similar microbiota will tend to cluster together. And so the thing that should be immediately obvious is that the healthy ones tend to cluster together, and the C. diff, massively infected ones, tend to cluster together. So what they did here was that it was actually really interesting. They tried to alter the microbiota in a number of ways, starting from here. So there's 4 different colors, blue, kind of beige, or brown, green, and gray. Each of those represents a different treatment. So let's start with the sort of most straightforward one, the healthy input. This is a transfer of fecal-associated bacteria from a healthy mouse. Everything. So all of the bacteria that were living in the gut of someone else brought into this mouse. And what happens? Well, here's before they do that. Here's after 3 days, here's after 4 days, here's after 6 days, here's after 14 days. So the mouse are cleared of C. diff, and you can see that in terms of the microbial composition. What about the other ones? Well, let's look at blue. Okay, so this is the same colony, but it's been passed through broth overnight. Okay? Purifies it a bit, might reduce some things that are less culturable. You see the same effect. Pre-treatment, 6 days, 14 days. We're back in this sort of happy land. Right? What's the other ones? Well, green is mixed B. This is where they took 6 specific organisms. So not the 30 or 40 that you might ordinarily find, but they took 6 of them, okay, that seemed particularly promising. And what happened? Well, here's pre, here's 4, here's 6, here's 14. So that mix of a much simpler collection of microbes actually worked almost as well. Okay? Well, that's interesting. That may be good for a few reasons. Would you rather have 40 microorganisms going into you, or 6 that are relatively well understood? So this is good to know. The last one I want to show you is the gray set of samples, where we have pre, 6 days, 14 days. They go off somewhere else. What is this treatment? It's actually Bacteroides and Lactobacillus. So your Activia, for instance, typically has 1 or 2 of these as probiotics, right? And what you see is that, sure, it altered the state of the microbiome, but it did not restore this sort of characteristic profile of a healthy microbiome. And so moving from mice to humans, one thing that's really interesting that I didn't know until recently, fecal transplants have been in the media quite a bit lately, right? So you think, okay, this is a fairly new idea. Well, actually, does anybody know when the first fecal transplant was described in the literature? Any guesses?
In the literature or generally?
In the literature.
Okay. They used to do them in China.
Yeah. Well, here's your answer, okay? The song of the year was Volare. Diefenbaker was prime minister. Michael Jackson was born, and Algeria was still a part of France. 1958. This was quite a surprise. There was a systematic review done in 2011 of this and other studies where they found 317 individuals, and they could collect reliable data, treated various ways, whether it's an enema or a nasojejunal tube, for instance, different ways of introducing the microbiota. But what was consistent was that the success rate in terms of resolution of these infections was usually about 85%, and the average is 92%. regardless of male or female. There were differences, but still in this range. And then there was a clinical trial, which many of you are probably familiar with, published just last year in the New England Journal of Medicine, where 15 of 16 patients with this really intractable C. diff, right, recurrent C. diff, within about 2 or 3 days were cured, right, because of the fecal transplant. And the clinical trial was stopped at that stage, and they offered this to the control group, and the control group did extremely well too. So this is quite promising. And so one interesting thing, and you saw this with the mix B, so here's an example of another paper last year where they said, instead of just taking this bunch of stuff from a fecal sample, which might have, bacteria that are not so wonderful, might have some viruses in it, right? Might have all sorts of stuff that we're not interested in putting into a sick recipient, right? Instead of doing that, let's isolate a number of different bacteria from, from genera that we know to be beneficial in most cases, and then put that into a synthetic mix, right? And so they call this the repopulate mix, of course. And the original paper was a trial of 2 patients, so not a large set, but they saw the same thing. They saw this sort of remediation of the C. diff infection within a couple of days, right? So this is very interesting. I mean, it still doesn't necessarily reflect on the fact that we don't understand precisely the ecological role of each of these, but it does tell us something about the potential of putting these things together. All right, so now I want to talk a little bit about the process of how we try and get to grips with the microbiome. How do we understand what's going on? Okay? And so the general procedure, at sort of a very high-level view, is that we start with an individual, right? Green silhouette man. Okay? And so the first thing we're going to do is we're going to choose a body site on green silhouette man that we want to focus on. Often this is the gut, for the reasons that you've seen already, but people have done skin microbiomics from all over. People have done oral microbiomes, nasopharyngeal microbiomes, lung microbiomes, you name it. Okay? Every body site, I think, that's not aseptic has been sampled. So then you have a sampling technique, right? It might be a fecal sample, might be a Q-tip, might be anything like that. And so now you've got kind of your raw sample from the human subject. Right? A couple of things you can do now. One is to bring this into the lab and start to do experiments, right? To really try and understand better what these organisms can do. You culture them, you feed them different things and see what happens. Maybe you put them together with another bacteria and see whether they fight it out or whether they have some sort of positive association. Right? So this is the experimental side. The other thing you can do is you can extract the DNA directly from your sample. Right? So you extract the DNA. The next step is to do some sort of high throughput sequencing. And actually, sorry, not just sequencing. You can do DNA sequencing, right? Genomics, metagenomics. You can do transcriptomics, you can do proteomics, and you can do metabolomics, which will reveal to you the small molecules that are involved, that are present, right? Such as butyrate, for example. Once you have this large data set, and you'll see how large it can be in a moment, then you do some bioinformatics with your cutting edge IT technology, information technology. And then from there, from these 2 sources of information, what you really want to know is diversity, right? Who is there? Function, what are they doing? Who is doing what? And then the big question of how will they respond when we try to do something to them? And what will the impact on the host be? So this is the general procedure, but there are a few challenges along the way. For example, first of all, you need to be sure that you're looking at the right area of focus. So I've talked so far about microbiomes that have been sampled through fecal samples, that have been sampled through fecal feces, right? And so the problem with that is that it's kind of a non-random sample of the bacteria that you'd find in the gut, right? If you imagine the sort of express train through the intestines, some stuff is going to be more readily picked up by this nascent bit of feces than other things are. In particular, you've got the lumen and you've got the mucosal surfaces, and you tend to get more luminal stuff because the mucosal stuff tends to be in closer association with colonic cells. So you might want to target one area specifically. Right? That can be challenging. The other thing is, even once you overcome that, you will have what I call the great microbiome discrepancy, where you have your in situ microbiota. That's what you want to know about. You'll have your sample microbiota. And what's the difference between those two, right? Do you have a good sample of stuff, or are there certain things that are over or underrepresented? How does that relate with the microbiota that are culturable? One thing that's interesting, that actually stands out about the human gut, is that many more microbes than you would typically expect can actually be grown in the lab. So that immediately tells something about the lifestyle of these organisms. Right? Why is it that they can grow much more simply than organisms from many other environments? What is that telling us? And then the last question is, to what extent can we take the data that we generate from a microbiome sample and actually analyze it in a way that doesn't just tell us a pack of lies, right? How can we computationally assess these things? And so the computational assessment is actually a really big question, a really big story from this, because ultimately what you get is a massive set of DNA sequences if we're talking about metagenomics, okay? And so there's a couple of different forms that this type of data can take. And so if you look here, here's your DNA sequences. They tend to be quite short. And you can either profile the microbiota using a conserved marker gene, right? Typically something that's present in all bacteria, such as the 16S ribosomal RNA gene. That's one way to do it. So you get information about phylogenetic diversity, but it doesn't tell you much about function, okay? The other thing you can do is just do a random shotgun sequence of the entire sample, and that can tell you more about the functional genes that are present. So if you've got a butyrate kinase gene that's present in 5 different organisms from your sample, the shotgun approach, proper metagenomics, stands a chance of telling you this information. So how do we put the puzzle back together to address the questions that I raised before? And so now I just wanted to touch briefly on the research, some of the development that's gone on in my lab, because as I said before, we really focus on computational approaches to make sense of these things. And so the first technique that I wanted to highlight, which was developed by a number of people in Canada and the US, but led by my former postdoc, Morgan Langell, who's now a professor in pharmacology here, was this method called PyCrust. Well, what does PyCrust do? I just told you about the difference between marker gene surveys, which tell you very little about function, and metagenomic surveys, which can potentially tell you a lot about function. PyCrust attempts to bridge the gap between these. What you do is, if you have a marker gene survey, and let me focus in because this figure is really busy. If you have a marker gene survey, you'll have, let's say, 16 S genes from a number of different organisms. How can you infer the function of the organisms if you don't know what they are, right? If you don't have sequenced genomes? The answer is, here's our sample sequences, so in gold. The blue ones represent sequenced genomes where we have the marker gene and we have the complete genome such that we know the functional information associated with this marker gene. What we're looking for, simply put, is sequenced genomes that are quite similar to the environmental genes we observe. We try to infer some sort of ancestral gene content and then map that into our mystery genomes. We've done extensive validations of this, and it works surprisingly well, actually. People are starting to use this on a big scale to try and get the functional information where it normally would not be available. Another one that we've developed, and so this applies specifically to shotgun sequencing, where you have these short DNA sequence reads, and you don't know what organism they came from, and you don't know what they're doing. So the function is one question, but assigning them to the originating organism is another problem. And so what do? Well, we have this rapid identification of taxonomic assignments where you feed the method a set of shotgun sequences. What it does is it takes a reference database, again of sequenced genomes, and it builds models from them. It says, oh, this genome has this copy of the butyrate kinase genes. Its genes tend to have more GC, sort of guanine and cytosine content relative to some other genomes. Rita attempts to capture this information quickly in order to make a prediction. And not just a prediction, so it would say, oh, this short sequence is probably from Faecalibacterium because the gene looks like Faecalibacterium genes, and the best homology match is to something from Faecalibacterium. Okay, I'm pretty confident in that. But if there's disagreement between the different lines of evidence, then it'll put that into another category and say, well, maybe take a closer look at this because we're not quite sure of it. The last one I wanted to mention is a more general tool that's mostly been applied to microbiomes. Okay? So if you have, and we'll keep it simple, 2 different samples, You have two different samples, and you have some functional profile of the two samples. So the first sample has these genes in certain abundances. The second sample has the same list of genes, but in different abundances. So there are different statistical tests you can apply to this, right? Such as the hypergeometric test, Fisher's exact test, and so on. This method, STAMP, statistical analysis of metagenome profiles, was developed to allow these comparisons to be done in a rigorous way, using interactive visualizations where you can actually rank these things, these different functions, such as metal resistance, in terms of their associated p-values, the p-values of their differences with multiple test corrections, or in terms of their effect size, right? Because if the p-value is 0.0000001, but the difference in frequencies is like 0.01% versus 0.02%, maybe you don't care so much about that. So this allows you to consider both of these factors in terms of making calls about what is most interesting. But we come back again to this question. We can do this. We can do taxonomic mapping. We can do functional mapping. We can try to figure out who's doing what. But it doesn't, it gets us towards, but it doesn't get us to this big question of what is every organism doing? Right? We can express it in metabolic terms, but what about the overall ecological role? Do we have symbionts, right? Do we have commensal organisms that contribute nice stuff to us? Okay? Do we have organisms that are parasitic? Sure, but how are they parasitic? One neat hypothesis, which is developed by a fellow named Mike Surratt at Master University, is that there are organisms called synergens, and he's actually demonstrated this in the cystic fibrosis lung. Here is a microbe. The microbe is neutral to us. It lives in us, but it doesn't really do anything good, or it doesn't really do anything bad. Or in some cases, it can be slightly beneficial. So that's microbe A. Right? Hanging around doesn't bother us. Introduce microbe B, which may be a pathogen, may not be. Microbe B, its presence, induces a response in microbe A that turns it into a pathogen. So the synergen is the organism that looks nice, right? Dr. Jekyll, but then brings some sort of stimulus along and it turns into Mr. Hyde and can have serious negative consequences to our health. Another possibility is that it's a passenger. Right? One important thing to know is that I talked before about species and genera as if they're things, right? What is the ecological role of Fecalibacterium or Prevotella or whatever? The fact of the matter is some of these genera and species are actually, they're differentiated at very fine levels. You can have two very similar strains that coexist. They're probably doing something subtly different. And those subtle differences might actually map into huge differences in terms of their impact on us. Okay? There's also the question of the interaction with the host genetic background. So those questions people are starting to tease out as well. And so now I want to move into an area that is of particular interest to some of us at Dalhousie because there is existing expertise in this. Dal or Halifax actually has, I think, the highest concentration of gerontologists in the country. And so there's a lot of interest in what happens to us as we age and the distinction between aging well, right, retaining a high level of cognitive function and mobility and so on into relatively advanced age, versus aging less well, right, which corresponds to the notion of frailty, right? And so this individual, you may have heard of him, Fauja Singh, right? He's running a marathon. This is about 2 years ago. He's 101 years old, right? So most of us won't make it to 101. The remainder, the one lucky one of us from this room, if there is one, ain't going to be running marathons at 101. So what's special about him, right? Well, it needs to be some really unique combination of his genetic background, the environment he exists in, his lifestyle, right, and maybe the microbiota as well. This is not a frail 101-year-old, right, unlike most 101-year-olds. So what's special about him? So we're not sequencing his feces. We're doing other things. So let me start though at the other end, right? The cute drinking milk, burping, getting diapers changed. So yes, exactly. So what happens at the beginning, right? My first microbiome. At the beginning, at birth, the gut microbiome is sterile. There's nothing there. As you start to grow and develop in your first few months of life, typically you're colonized, you drink your mother's milk, you're typically colonized mostly by bifidobacteria, which is a group of microorganisms. Never mind the details. One crucial thing that happens at this stage is the maturation of the immune system, right? So that leads us down certain roads where you say, well, What if the bifidobacteria that are supposed to be there aren't there? What does that mean for our immune system, the development of allergies, and so on and so forth? Around the age of 2 years or so, and so by the way, this is richness. This is the number of, finger quote, species, okay? So you can see this increasing diversity. We reach an age of about 2, and then we start transitioning into a more adult-like microbiota, which is dominated by 2 groups, Bacteroidetes and Firmicutes, very specific members of those groups. So there's sort of a reverse riddle of the Sphinx, right? So one of the riddles of the Sphinx is what has 4 legs in the morning and 2 legs in the middle of the day and 3 legs at night? And the answer is man, or humans, right? Because you crawl and then you walk with a cane, right? So the reverse of that is we know that it's a man or a human, right? And we kind of know what they have early on in the morning. We have some sense of the possibilities, in the middle of the day. But what about in the evening? Right? Jeanne Calment celebrating her 121st birthday. What happens here? Is there a story that's as clear and consistent as we see in infants and young children? Not really. It's a much more complicated story, but there are some trends that have emerged from studies of aged populations in Ireland, Italy, India, and possibly other countries that start with the letter I. So you see some things like this very important commensal, Faecalibacterium prausnitzii, which ordinarily has an anti-inflammatory role. It's one of our best friends, these butyrogenic organisms, and it goes down. It tends to go down quite a bit. In addition to that, Clostridium cluster 14A, what on earth is that, right? Thanks, taxonomists. Which comprises actually several, again, butyrogenic organisms, actually goes down as well. And then we see increases in things like strep and staph and other facultative anaerobes. So immediately warning bells go off, right? Does aging tend to lead to a increase in dysbiosis? As we lose good things, we gain bad things. The fact of the matter is that these are trends, and what you see in individuals is all over the map. You can look even just at the balance of Bacteroidetes and Firmicutes, and it goes from 5% to 95%. So this tendency is certainly not repeated in everyone. What about frailty? Well, there's less work that's been done in frailty, but there's nonetheless a bit of that information out there. So one thing is the drop in another group called Bacteroidetes. These can actually have both beneficial and harmful effects. Okay? So this is a bit less clear. But then we see an increase in certain markers of inflammation, things that are non-butyrogenic. Some of these produce lactic acid, for instance, which is less good for us. These people have found associations with things like strep, between strep and then some sort of cognitive or nutritional deficits. Okay, so there's some specific trends that people claim to have found that might relate to specific organisms and specific conditions, clues as to the roles. Right? And so there's this whole, these ideas of inflammation as you age and the senescence of the immune system. Is that connected to dysbiosis? Maybe. So here's what we're doing right now. Okay? We have a pilot project going on, which is looking at fecal samples not from humans, but in fact in mice. Okay? So Susan Howlett and Rob Rose in the Tucker building have mouse colonies, and they've developed quantitative models of mouse frailty. So when we have a mouse, we can quantify its age, which is not hard. We can quantify its frailty based on a number of different tests that assess cognition and behavior, and so on. Okay? So the basic idea of what we've done so far is taken 21 fecal samples from mice of 3 different age groups. Okay? And I'll tell you right now, we're not able to tease out frailty from this because all the young mice are not frail, all the middle-aged mice are slightly frail, and all the old mice are really frail. These are 2.5-year-old mice that are literally at death's door. Okay? Don't ask to meet them because they're not there anymore. So what we did, we took these 21 fecal samples, and then using a Illumina HiSeq from Genomics Quebec, sequenced back. We got 214 million DNA sequences collectively, 42 billion nucleotides, which is equivalent to about 14 human genomes. That's the data we have to work with from this small pilot study, which cost us $6,000. Okay. Hopefully this underscores the magnitude of the problem. And at the bottom is just a little bit about how we need to use super efficient methods that don't always guarantee the best answer. Because if we used the more precise approaches, in this case, if we use BLAST, for instance, basic local alignment search tool, the sort of gold standard, or almost the gold standard for homology search, it would take about at least 50 times longer. Time we don't have. Okay? And so here's a brief summary of what we've seen. And the first thing to notice is we have the young, middle, and old mice separated out. And the bars here correspond to different groups of bacteria, right? Again, what are the identities? Well, that's a hard thing to pin down. These are bacterial families, by the way. Above genus, below order. Okay, what does that mean? And so you can see massive differences. You can see huge differences in the relative abundance of Bacteroidetes, right? Very few, quite a bit, and even more, right? And so you can look for that. You say, well, can we tease out any trends, right? And there's a couple of interesting things here. One is that we have this green group which starts to come up in middle age and then is present but in slightly less amounts in the older mice. Okay? And this is actually the group Recanalaceae, which includes allostemps, which is one of those markers of inflammation. Okay? So that shows an interesting pattern. Another thing we see is that this orange bar at the top tends to go up. Okay? This is Prevotella. Again, another organism that is not necessarily helpful to our existence. This is, again, a principal components analysis plot, and all I'm saying here is that we don't tell the PCA what the clusters should be, what the groupings of these organisms, these samples should be, but it finds them anyway, because they do have really important similarities in their taxonomic composition. That is why we happen to get the middle-aged group here, and the old group here, and the young group there. I'm happy to talk about the gory details of any of these methods, but hopefully at least the main outcome is clear. We can look at specific functions, because this is a proper metagenomic study. We use not just the marker gene, we also use the functional genes. And so one thing we find, and these are not quite properly arranged, green is young, blue is middle-aged, and orange is old. What is the gene? What is the function? Creatine degradation. Okay? So creatine is very important to us, but the bacteria associated with these mice carry out a lot more, or probably carry out a lot more creatine degradation as these mice are getting older. There's a number of other functions that we found to be interesting, and it's just a matter of trying to put a story together, right? Because there's, you have a number of functions. What do they collectively tell us? And so the next steps in this, we've got our mice. Now we have a little bit of funding to start piloting this in older patients, in frail populations, specifically in an assisted living center at first, but then looking at other settings such as day hospitals, specialized clinics, people just like community living individuals, right? Because the contrasts between these individuals will be very important, very informative. Especially as we get into larger sample sizes, because with that amount of variance, it's very difficult to get a consistent story. So I wanted to finish up with a couple of things. One is sort of the general directions where we need to go in order to understand the microbiome in ways that will be clinically useful. The other thing is that we have a couple of projects. In fact, Dalhousie has a few projects starting up related to microbiomics. So this is an area that's really gaining interest around here. People are initiating new projects, there are new faculty members being hired in the area. So it's kind of nice to know what's going on. First though, there's a few things we need to think about. One is to sample and assess more broadly, right? And so one part of this is, as I said before, try to understand the difference between mucosal surfaces, let's say in different parts of the colon. Are there differences? Well, as you can probably imagine, now we're not talking about fecal samples at all. You need to actually get in there to get specific samples from different parts of the colon, right? So that's part of it. Another one is actually what's called multi-omics. People are starting to do this more, where you don't just do the genomics. The genomics tells you about the potential. The transcriptomics tells you about what genes are being expressed in a given condition. Right? So if you're profiling at the wrong time, you may not get the information you want. Proteomics, similar idea. Metabolomics is particularly interesting because it tells you what molecules are floating around, which can help or hinder the other bacteria, as well as the human host. We need new models, new ways of expressing the interactions between the sort of three-cornered triangle, where we have the host, we have the microbiota, and we have the environment. How do these things relate to each other? If we perturb one, what happens to the rest? A couple more things. I've said this already several times. Oh, you've got Prevotella at 10%. What? What does that mean? Does it mean several things because there's Prevotella this and Prevotella that and they have different roles? Right? What is going on here? If Prevotella goes up, what does that mean for you? And what does that mean for the other bacteria that live inside you? That's a very difficult thing to do. People have looked at it just from the sort of observational point, but we lack the first principles models to really get at this. And the other thing that's really important is that I've talked a little bit, but sort of lied in the details about species or things that are like species. But the fact of the matter is, we don't know the right level of resolution to consider. In some cases, it might be the strain. This very specific strain or isolate has a very specific role to play. In other cases, it might be the species, and every member of that species does more or less the same thing. Genus level, right? Maybe that's what's important. Okay? Different species in the same genus, doesn't matter which one you have, right? One thing that's sort of near and dear to my heart, which has really turned up in quite high amounts in the human microbiome, particularly the gut microbiome, is that the bacteria in there share genes like they do in no other habitat on Earth. So there's gene sharing that goes on in the environment, right? In hypersaline organisms, thermophilic organisms, in dirt. But none of those rates of LGT can even come close to the rate of lateral gene transfer in the human gut. What does that mean? What's going on there? What's the role of lateral gene transfer? So a couple of projects to finish up. So this is one that got kicked off a couple of years ago under the leadership of Fort Doolittle. For short, we call it the it.The project. And the basic idea here is not so much to collect new data, we're not so much doing sampling and sequencing, but it's really about trying to understand the microbiome and translate that understanding into new methods that really get at some of these challenges. And so PyCrust, which I showed you, is one of the outcomes of this, right? And then there's a philosophical component where we try to get at these issues of defining concepts, right, which can help us to really get at the units of diversity in the microbiome. And also the question of interactions in the microbiome, because obviously not everyone is just playing by themselves. There's stuff going on at a more complex level. A couple examples. This is a very colorful figure that's also very complicated. All I want to say about this is that this is a method developed by Madi Shafieh and Joe Belowski and some analysis by Eva Moon. And what's going on here is they have samples from 4 different groups of people. And I believe this is from the Irish study. Everything that's done in this method is unsupervised, which is to say the method never says, this is the metabolic pathway. It never says, this is the group that you're trying to classify. It is totally up to the method to figure these things out for itself. So what happens is the method says, oh, this chain of reactions really seems to differentiate some of our samples. It doesn't have to be a cohesive pathway. It could be 2 pathways, half of a pathway, doesn't matter. Right? Similarly, once you have these models of different, sort of metabolic units, you can define things at a higher level. It's called a metabotype, and then see how your samples, these different metabotypes comprise your samples. And you won't necessarily see that, this sample is entirely metabotype 1. That's why these things are not all kind of cleanly at the corners of these triangle. But you see these mappings, and what's interesting is that the infants and youth have certain metabolic functions that distinguish them from the other set, even when you don't tell the method what they should be. So this is a really neat method. Connor in my group has been looking in detail at some specific groups of bacteria that are found in the human gut, in particular a group called the Lactospiraceae. They're in class Clostridia. Usually when you hear Clostridia, you think, ugh, right, it's C. diff, but the fact of the matter is, many butyrogenic organisms are in the Clostridia. Long story short, this is a clustering of a number of different Clostridia based on their gene content. Okay? If their gene content is similar, they have the same repertoire of functional genes, they'll be adjacent to each other in this clustering. And so the question here is, can we get at some sort of ecological similarity? One thing that's quite interesting is that usually when people talk about Lactospiraceae, they talk about beneficial organisms, they synthesize butyrate. But what we get when we look here, and I just specifically want to draw your attention to the blue elements of the cluster, because the other ones are not gut associated, that's a different story. But basically, if you look at the blue, then there's this cluster of human associated, And then there's another cluster of human-associated organisms on top. These differ in certain key metabolic functions. This cluster contains all of the deuterogenic organisms we know about. This is a poorly characterized group we know less about. So what is the difference? What is going on here? We don't know. Okay? The other thing about Clostridial taxonomy is that it is a mess and it doesn't help. Speaking of lateral gene transfer, the deuterogenic organisms in this group appear to have shared butyrate synthesis genes quite a bit. So this crucial function is actually getting passed back and forth between different organisms. By contrast, an important function such as sporulation, which is present sporadically throughout members of the group, does not show evidence of lateral gene transfer. It's been lost in many lineages. A couple of other projects, just very quickly. This is the complement pathway induces certain types of responses that kill pathogens, right? And so Andy Stadnick, excuse me, has identified some activators of this pathway and then generated mice that are deficient for these activators. The key question is, do these mutant mice, do they recover in a similar way to colitis recovery? Do they recover from colitis in a manner similar to wild type? Or is their recovery compromised by their inability to activate this pathway? So we need to look not only at the phenotype of the mice, but also the microbiota. Excuse me. Last project, this is one I'm not involved in, but it's pretty exciting. So Johan van Limburgen is a new faculty member in gastroenterology and nutrition. And they have a big study that's just starting up where they're going to look at Crohn's disease in children, right? And focus on specific parts like the mucosa, and then try to do what I showed you with the triangle plot there to see whether individuals can be separated in terms of their metabolism and their microbiome, with a mind to better understanding the role of the microbes in pediatric Crohn's disease. So, that's it. I hope that I've given you a reasonably good overview of what's going on. I don't expect you to remember every genus and species name I've given you, but the idea here is that they're diverse, and we really do not understand what a lot of them are doing. Nonetheless, there is a path towards greater understanding, and it involves experimentation, and it involves a lot more bioinformatics. And so that's the objective, right? And that's where we're hoping to be in the next 5 to 10 years. So with that, I'm really finished, and I would be delighted to take any of your questions. Thank you very much.
Just a reminder, if you have a question, just raise your hand. I'll come over with the microphone. And if any of you have to leave, because we're just going to over 1 o'clock, please feel free. But we won't have questions, I'm sure.
Thank you. That was a terrific talk. I wonder if there's been opportunity yet to look in family groupings to see whether sharing of the close environment, perhaps in couples where you share a kiss or something like that, that the microbiomes then become similar, and whether that has any role to play in the health of the individuals.
There's a couple of interesting studies that come to mind. So one is more on the sort of host background genetics, and it's about mothers and their children. And long story short, during the period of breastfeeding, I think the microbiota of the infant really shows its sort of origination from the mother. But after weaning and the transition to solid food, the kid typically develops its own microbiota, which may not be connected with that of the parent, but the mother. Another really interesting study, which was done by Rob Knight, pretty much, Rob Knight does a lot of studies. This one was on the microbiota of people living in the same home, with or without pets. And so it turns out that, yes, your dog can influence the microbiota that you have.
And the connection to health?
Oh, the connection between host background and health, or environment and health? Well, that's a great question, because basically you're asking, is there a familial predisposition to certain types of disease that is transmitted via the microbiota rather than through the host genetic background? And I don't know the answer to that. When there tends to be somewhat less correlation with the microbiota, I'm not sure whether the genetic structuring of the microbiota we might expect would have an influence on that. But Connor has his hand up.
I can help with that. Yeah.
Go ahead.
So they've done monozygotic and dizygotic twin studies on it and found that there isn't as much of a correlation with the genetic side. But a lot of studies say that people who live in the same household have very similar microbiomes to each other, and that's true, but it's probably due to the environment more so than specifically the genetic one. But there is still some genetic dispositions in people to have certain species or strains associated with them in the microbiome. So it's still kind of up in the air, but it's influenced by so many different things. Like there's studies that show that depending on the plants that you have in the house and the plants that you have in your garden will influence what the microbiome of the kids is. So there's 500 different factors when you're trying to tease out one of them. It's still a little bit difficult to do so.
I would be very surprised if human genetic variation that impacts, for instance, on the colonic cells does not have an influence on the microbiota structure, which in turn, one would expect to have an influence on the health of the host. But as far as I know, there's very little that's been done in this way. In part because the sample sizes tend to be in the dozens or hundreds, and as Connor says, these types of effects, you might need thousands or tens of thousands of people to be able to sort them properly.
So to get around that problem, people done a lot of the microbiome work on mice and different genetic backgrounds of mice, different diets, and the same genetic background of mice to try and get rid of as many of the environmental factors as possible. So you didn't come in.
So basically, you just have the genetic factors or the environmental factors. I mean, the work I'm most familiar with is in notrobiotic mice, right? Where you have, maybe a diverse population, they're germ-free at first, and then you introduce humanized microbiota, right? And surprisingly or not surprisingly, this works. And certainly you can see environmental influences. But as far as whether the genetic structure of the mouse influences, well, actually there was that study. There was that study where they profiled the microbiota of 6 different widely used lab strains of mice. It was 6, right? Yeah. And yeah, we looked at this with our mouse study because we have a small sample size. We were wondering if we could draw on the hundreds and thousands of other mice that have had their microbiota done, like healthy mice, at sort of a particular age, 6 months a year. But it doesn't work because these different strains of mice, seemingly grown up in different locations, nonetheless have distinct patterns of microbiota. So, in fact, yes, the genetic background of the mouse does appear to influence the microbiota to some degree. And we did a clustering analysis, right? We said, okay, here's our 6 reference strains of mice from a previous study, and here's our mice. And the biggest factor by far was the strain of mouse, so that we could not, in fact, use other people's mice as an additional reference set for our study.
In humans, the biggest factor is diet. Diet will change it within one day. Your diet will change it.
It's sort of the same point, but you'd expect really interesting correlations between human genetics and diet and mice. I mean, particularly like lactose intolerance, which has been really well studied as a population thing, and correlation between the genetics of populations and the diets that they had, how much milk they used, obviously affects the microbiome too. So there should be kind of a 3-way interaction between culture, genetics, and microbiome.
Absolutely.
Questions?
That image you showed of the variation in proportion of bacteria over time, quite early on in the talk, and it just seems such an enormous challenge then to use that going forward. I wonder if you might talk a little bit about how you're thinking about tackling a moment in time versus a year's worth of variation, and how that will correlate with health of the individual and frailty.
Yeah, I mean, it certainly underscores the fact that a sample taken from an individual at one time point is not necessarily reflective of the overall composition of that individual. I mean, if you got this person on day 25 when the frequency of Preventalaceae was a great deal higher, then you might get a misleading picture of that individual. So, this is interesting from the point of view, for instance, I don't know if you're familiar with the study that talked about enterotypes of the human genome, or the human microbiome, where they profiled, I think it was 120 individuals, is that right? It was over 100 individuals. And what they found was that when they did some sort of clustering of the microbiota, kind of a clustering plus, they recovered 3 seemingly distinct groups of microbes. They were dominated by different microorganisms. However, if you took this one individual and sampled them at particular times, you might end up with 3 clusters as well. So there's a few problems with the enterotype hypothesis. This is one of them, right? And so what do you do about it? Well, I think this is where models become important, right? If you have some sort of model of the relationship between the microbiome and other stuff, whether it's diet, whether it's fever, whether it's any other sort of factor, then I think the strength of your ability to do these sorts of, interpret these sorts of things would be much stronger. Because, the microbiome can change in hours. It takes very little time to administer an antibiotic, and that's going to have almost an instantaneous impact on the microbiota. And so you cannot possibly sample, or at least, it's very difficult to reasonably sample from hour to hour, for instance, and try and get that really fine-grained stuff. But if you have, these broader correlations that you might get from a reasonable set of studies, that might do a much better job of allowing you to drill down and really try to understand what's going on here, right? Why is this red? What does it mean?
I'm stepping on your toes here, answering things, but Joe Golewski's group, who were building that other model with the 3-sided thing, they worked on this data, and actually, even though you get these kind of stochastic changes over time, I think they can take 15 days worth of samples and actually predict the person still. So you get these changes over time, but if you have a couple of days of samples, you can tell one person apart from the other. And so that there is still some kind of signal, even though we see a lot of changes. And the other thing that we work in our group is that this is changes in species, but it might be that you have a functional core that's the same. So it's like, you get massive changes in the number of species, but if all of them are producing butyrate, the function stays there for the microbiome, and then just the people who are doing it around it change. So it's about switching it from more of a traditional ecological view of like, this species must be here to do this, and more this set of species must be here to do this particular function.
I think one interesting story that I didn't really touch on in my presentation was this sort of duality that many people talk about where, as Connor said, the taxonomic variation of the microbiome can be large, and the functional variation of the microbiome can be quite small. But if you look at the basis for that assertion, for instance, if you look at the big human microbiome project paper where they did a huge amount of sequencing of a massive number of individuals, they say, the function is conserved. But when you look at the level of resolution of function, if you're profiling the function of thousands of genes, you necessarily must summarize them at a very high level, whether it's metabolism or certain specific types of metabolism or certain types of receptors. And the simple fact of the matter is the stuff that matters, the stuff that changes and has an impact on the human host, might be a lot more subtle than that. Right? A specific enzyme in a specific pathway can make the difference between synthesizing butyrate and having just an intermediate product. And so if you look at these large-scale summaries of function, then, and sometimes the function is like ribosomal proteins. Well, guess what? That's not going to change no matter what you do. So I think that one other crucial piece of information will be, one more thing about that. If you've ever looked at a bacterial genome, you know that anywhere up to a majority of its genes are of unknown function. So never mind the summarizing at high levels, there's all this stuff we don't know about, which might be involved in ecological interactions, defense, attack, other interactions with the host. We don't know any of that. So how do we get at that finely detailed level without entering sort of a statistical purgatory? That's another important question, to answer your question with a question.
think we may have to call it quits, unfortunately. But I guess it's not surprising why this was the most subscribed seminar we've ever had, because it was fascinating. So thank you again, Rob, for being here. Let's thank you again.
Thank you, everyone. Drive safely.
Before you go, I just wanted to mention that we have this seminar series is CME accredited, and we would really appreciate it if you would fill out your evaluation if you want to get one of the credits. But we'd also really like for you to fill out the evaluation even if you're not, because we're really curious about how effective we're being with the seminar. We've revamped the evaluation to get some deeper feedback so we can make these what this community needs. So please do that on your way out. And in February, February 19th, we have another really great session coming up with Deborah Marshall from the University of Calgary, who is a health economist, and she'll be talking about the role of looking at the economics of health from a personalized medicine perspective. So that'll be a really interesting one for, I'm sure, everyone in the room and more. So make sure I'll be sending out the invitation for that shortly, and make sure you register early. All right, thank you again for coming. Be safe today.